Hi, I'm Prachi Joshi. I'm a junior research group leader in geomicrobiology here at the University of Tübingen.
Hey, I'm Nora Kainz. I'm a PhD student in the Coastal Wetland project, which we are going to introduce to you today.
Climate change is caused by increasing concentrations of greenhouse gases in our atmosphere that warm up the planet as well as have cascading effects. Two of the most important greenhouse gases are carbon dioxide and methane, and part of the release of these carbon dioxide and methane gases into the atmosphere comes from natural sources like soils and sediments. In these systems, microorganisms slowly decompose the remains of plants, animals, and other microorganisms to release greenhouse gases. And this process is particularly important in wetlands that serve a dual role in the carbon cycle by acting both as a sink for carbon as well as a source of greenhouse gases. In this project, we focused on wetlands of the coast, so the interface between the land and the sea. An example of this would be salt marshes, or if you're in tropical regions, mangroves. In these systems, organic carbon gets accumulated, we call this carbon blue carbon, through the trapping of incoming particles and by the growth of plants. The input of organic carbon to these systems is delicately balanced with the output as greenhouse gases. And we know that this balance is very sensitive to disturbances like rising sea levels, increasing temperatures, and changes in land use. Our goal is to understand what controls the release of greenhouse gases in coastal wetlands today, so that we can better predict what will happen in the future. We do this by studying the release of greenhouse gases in these ecosystems and combine lab-scale and field-scale experiments. The reason we do both is that lab-scale experiments really allow us to zoom in on the most important processes while also being able to control external factors. We do field experiments because it allows us to incorporate all of the complexities of the real world.
For this project, we worked at the north coast of Germany at the Wadden. In summer 2023, we did a large field experiment to understand the greenhouse gas release through microorganisms. We did this by injecting organic carbon into the sediment, essentially giving them different types of food and looking at how much CO2 they release in response. To do this, we set up small plots across the field, into which we injected organic carbon. We then measured gas emissions by using flux chambers, from which we took multiple gas samples after 1.5 hours, 24 hours, and again after 48 hours. We also looked at the effect of microbial activity by looking at the chemical species in the pore water as well as in the sediment. For this, we sampled pore water after the last gas measurement. And at the end of the experiment, we also took sediment cores. After the summer, we brought the samples back to the lab and analyzed them. From this analysis, we aim to understand if the microorganism can use the injected organic carbon and if this results in a higher CO2 emission. This allows us to predict how the ecosystem will react with increased organic carbon inputs, and it's important to better understand the carbon dynamic in these ecosystems.
Blue carbon systems, like the salt marshes that we study, are crucial parts of the global carbon cycle. And understanding this delicate balance of carbon inputs and outputs in these vulnerable, threatened ecosystems is critical to how well we can predict greenhouse gas release in the future.